Hello friends, welcome to BMH Learning. In this video, we will talk about RAD-seq, also called Restriction Site Associated DNA Sequencing. RAD-seq represents an alternative to whole genome NGS sequencing for the detection of SNPs and indel variations in DNA. This method consists of the targeted sequencing of regions adjacent to common restriction sites distributed throughout the genome. The use of several restriction enzymes will increase the coverage of the genome without the need for a reference genome. How RAD-seq works? RAD-seq works by first fragmenting the target genome using a restriction enzyme. After digestion by a restriction enzyme, a first ligation allows to add to the DNA fragments a P1 adapter, complementary to the restriction site and barcoded for sample identification. P1 also contains the sites of the amplification and sequencing primers. The next step is a mechanical fragmentation associated with a size selection of the fragments according to sequencing requirements. The P2 adapter is then ligated to the selected fragments. PCR with the amplification primers P1 and P2 will allow amplification only of the fragments carrying the adapters P1 and P2 at their ends. These steps transform the DNA into a fragment library suitable for sequencing on an NGS platform. Depending on the end goal, either single-end or paired-end sequencing can be used to generate the appropriate amount of information. Sequenced data is then analyzed to identify and score genetic variations in the samples or population of interest. This was all. Thanks for watching.